Okay. I'm in.
You're in.
Okay, good.
Hold on, let me introduce you. So our next speaker is Dr. Ammasi Periasmy, affectionately known as Perry. He is professor of biology and biomedical engineering at the University of Virginia and director of the famous Keck Center. Perry is one of the pioneers in the development of FRET-based imaging and has held many FRET workshops over the years. During the pandemic, I asked Perry how he's been keeping himself busy in addition to everything else. And he said he's taken up cooking spicy Indian dishes and running 4 miles a day to avoid the COVID gut. I will say, I think there's a lot of people who've taken up cooking during the pandemic, and it sounds like the kitchen is our wet lab when we can't actually get to the lab. So today he'll be telling us about his work in the investigation of metabolism and tryptophan interaction with NADH using single excitation FLIRR and FRET. So Perry, go ahead and share your stage or share your screen. Keep going in presentation.
It's not showing.
I see everything. We just need you to get the PowerPoint open.
I already opened the PowerPoint. Can you see it?
Yep. Now just go to reader
okay.
view. Yep.
Okay, sorry. I was looking like a Zoom.
Yeah.
Okay. I'm good. Okay, thank you. Thank you very much, Adila, for the nice introduction. And for the last 17 years, I was in Berkeley. This year, I am in my office in Charlottesville. Thanks for Holly and Wolfgang and Feather, Jens, for a nice organization. It's going very well. I have been watching since yesterday. Holly is doing a fantastic job organizing this. All right. So these are the people host, and Gupta is the main player in the NADH work. Kay is working on the cell culture. Rufan worked on the single wavelength for the redox ratio. Carsten is the data analysis. It's been very, very helpful for us from the computer science department. Jesh and she is, I always present something new in this workshop. Today I'm going to add machine learning. She is a graduate student. She just did a project in my class, but finally it became a paper. So that is quite interesting. And of course, funding from different NIH organization and the Cancer Center. This year, I'm not organizing the workshop because after Berkeley workshop and the SPIE meeting, my workshop is to be in March. I canceled this year. Next year, if everything is all right. So today I'm going to talk about, as Adila mentioned, that fluorescence lifetime redox ratio, which we introduced in 2016 using lifetime technique, which is two-photon, 2 excitation wavelength versus 1 excitation wavelength. And also I talk about the FRET between tryptophan and the NADPH. The new thing which I mentioned is the machine learning. About this, I will tell you later how we use the machine learning approach in the redox ratio or NADH measurement. So mainly our work is focused on prostate cancer. After 50 years or old, if you go to the doctor's office, he will ask you whether I can test your PSA, prostate-specific antigen. So but if you have the cancer, it will show up only if the PSA is 5 or 10 or something like that. It's not, will not show up at early stage. So we are trying to use this technique whether we can detect early stage. I am not going to show the clinical data, which we did some work on the PSA testing. We were able to detect up to 3 PSA. We are trying to improve going to the lower level. If there is, for example, for me, it is 0.4. So if you have any prostate cancer development, it goes from 3 to 10 or more. So that's what we are trying to focus on this work. So the CDC mentioned that the diagnosis and the cancer issue is going down now. The diagnosis is improving. The treatment is getting better. Now it is decreasing. As you see the graph, and the prostate cancer is also one of them. So you heard about a lot of talk about fluorescence lifetime imaging yesterday from Wolfgang, and also NADH and FAD work from Angelica and Marina and Elena. And now today you had a fantastic talk from Melissa Scala from about the stem cell. So I am also going to talk about the NADH for the prostate cancer work. So cancer metabolism, which is what we are measuring, is the optical redox ratio, which is a measure of the metabolism. Due to metabolic alterations, changes the coenzyme NADH concentration, and thereby the total NADH fluorescence intensity. Angelica gave fantastic talk on the flavin. Flavin exists predominantly in mitochondria and with the negligible concentration in the cytosol and nucleus. Is covalently bound in the mitochondria and only in the mitochondria complex 2. So these are the players. NADH FAD is involved in the redox ratio calculation. I am also going to cover the tryptophan, which is the important factor in the cancer progression. As you know, what we are measuring is the mitochondrial dysfunction. If any disease process, as it listed here, is basically involved, somehow mitochondria is affected. So you can always interpret your data by measuring the redox ratio in the mitochondria. So this is the system we used. It's a size 780 and 3 PMTs from Becker and Kickel. And this is customized system because the tryptophan emission is 300 to 400 nanometer. And so this is the UV transmission. Most of the microscope transmission is 400 to, if it is IR, 1200. So it's a customized system to measure the tryptophan. The green color is NADH. We label the mitochondria to demonstrate that what we are measuring NADH is from the mitochondria. So the optical redox ratio has been in the literature for a long time, introduced by Burton-Jones. You can do the optical redox ratio as fluorescence FAD over NADH or vice versa. Either way you can do. So we introduced the fluorescence lifetime redox ratio, which is we are ratioing the NADH enzyme bound fractions on the fluorescence FAD enzyme bound fractions. So this way we can, I will tell you why we are using this technique, FLIRR, which is now, Angelica compared this FLIRR technique very interestingly for different disease process. So if you use the Becker and Kickel software, you can see if you put two component analysis, A1 person, A2 person, A1, A2 is the pre-exponential factors. If you use some other software for data analysis, you can use the A1, A2 as the pre-exponential factor. Here it's listed as A1 person, A2 person. So why we went for the intensity, I mean, fluorescence lifetime redox ratio. So when we did the live animal for cancer studies, and we found out that it is jumping all over the place, normal versus tumor, it is maybe due to this is differential absorption of light or scattering in light in the tissue. So we used this redox ratio calculation using the bound enzyme of NADH and the bound FAD. You see that it is so stable output. So we realized that there is a optical light scattering issue, interference is taking place. That is why the distribution is not reproducible. So for the tissue, it is very important. For the cells, it's improved the sensitivity. I will show you some data over here. How do we apply this? First, we did the cell segmentation. That is where Costin and Host worked on it very good. We wrote a software so that we developed the software so that it goes 2 by 2 pixels and selects the mitochondria area in the cytoplasm. As you know, all the mitochondria are in the cytoplasm. So what it does is that the OXPHOS takes place in the mitochondria, the glycolysis takes place in the cytoplasmic. So this allows you to differentiate between glycolysis and OXPHOS. This is one thing. And number two is that you can do cell segmentation and able to produce, able to get the heterogeneous distribution of this redox ratio. So it's very, very helpful, very accurate, reproducible. It improved sensitivity of the measurement. So how do we test the sensitivity of the measurement? As you know, the cobalt chloride blocks the connection between the Krebs cycle and OXPHOS. So we added the cobalt chloride, about 50 micromolar, and overnight, and then we imaged the next day. You can see on the top panel is the cobalt chloride intensity, the left one, intensity ratio, and this is the FLIRR, ratio. You can see the arrow mark indicate increase in sensitivity of the redox ratio. We added the glucose, about 25 millimolar glucose for 30 minutes, and you can see the response. The middle graphs of the glucose shows the intensity, optical redox ratio. Below bottom is the FLIRR, the FLIM redox ratio. You can see the increase in redox ratio measurement sensitivity. And then we also investigated with the toxorubicin treatment for different timing. So you can clearly see the sensitivity ratio is increased. The heterogeneous distribution is clearly displayed using the FLIM technique. So it's a real experiment. Segupta did this work on LINCAP cells, which is very responsive for the drug, and African American prostate cancer cells, which is provided by the Dayan Chandra from Roswell Cancer Institute. And you can see the response for the drug is very good here. And you see the cells are, morphology of the cell is changing. I will come to that point when I go to the FRET work. I will tell you how the morphology changes. So we did for the two wavelength, the NADH 740 and the FAD 890 nanometer for FLIRR. So we've noticed that in neuronal cells, for we are working on Alzheimer's disease, in the neuronal cells, the mitochondria moves faster. Then we have spent 30 seconds to collect the NADH image, or 10 seconds, depending on the signal level. During that time, the mitochondria moves. And we thought that single wavelength will be an ideal situation. So Rufan worked on the project. And here is an example of due to disease process also, the mitochondria aggregates in some places. So it will be a nice idea to do single wavelength. Another second point is the crosstalk from NADH signal going to the FAD channel. So how do we control it? So this is another, the single wavelength helps to reduce the crosstalk. So here is an example I'm showing 740 nanometer. The NADH signal is less for 800 nanometer excitation. We have chosen the 800 nanometer, but we have enough counts, more than 1,000, depending on how long you collect. We have enough signal. You can see we used a solution to test this, 150 micromolar of NADH plus 100 micromolar of FAD signal, FAD solution. And you can see on the right side, the rectangular box shows 800 nanometer, the NADH, it's a normalized intensity, and the FAD signal, also it is very bright signal source. The crosstalk also reduced. You can see the 800 nanometer excitation, 1 hour, 2 hours. This is, you can clearly see for 740 nanometer versus the pattern, output pattern of the A2% remains the same, whether you use 740 nanometer excitation for NADH or 800 nanometer. The same case for the FAD bound A1%. The top panel is 800 nanometer. You can see the pattern. The dark line is control, and this is for 30 minutes and 1 hour and 2 hour time collection for toxorubicin. The same as for 890 nanometer. The image quality is not great, but for 800, the FAD image is very, very good. We get a lot of counts, photon counts for 800 nanometer for FAD. The FLIRR calculation, again, it shows the same pattern as 800 nanometer is on the top, 740 or 890 nanometer on the bottom. It shows the same kind of pattern. So 800 nanometer works pretty good. We are trying to use, and also it helps for time-lapse imaging. If you want to do time-lapse imaging using single wavelength, simultaneously you can collect both NADH and FAD. All right, so that is the optical redox ratio versus the FLIRR redox ratio. And now I'm going to show the tryptophan interaction with the NADPH. I have enough time. So using 740 nanometer, the 740 nanometer is a 3-photon excitation for tryptophan. When we use for redox ratio, the 740 nanometer is a 2-photon excitation for NADH. So basically, we use the same faster energy transfer principle. For FRET is a faster resonance energy transfer. Somebody is new here, listening to the talk. What is FRET? FRET can tell us about dynamic behavior of biological molecule and biological systems. And it requires 2 fluorophore should be spectral overlap. On the left side, you can see green and red. If the spectral overlap is there, then if you excite the donor molecule alone, the intensity increases in the acceptor fluorophore molecule. That is what we call as a sensitized emission. This is possible only when the distance between the molecule within 10 nanometer and the dipole moment orientation of the emission, donor emission to the acceptor absorption should be between 1 to 4. If it is perpendicular to each other, it is 0. So if you see a lifetime, how do you measure the FRET? Basically, you follow the donor molecule change in lifetime in the presence of acceptor or in the absence of acceptor. If you take a donor alone, this is the lifetime. If the donor is attached in the presence of acceptor, the lifetime decreases, or what we call as quenches. So you can calculate the energy transfer efficiency 1 minus lifetime of the donor in the presence of acceptor divided by the lifetime of the donor alone. There are a number of ways you can calculate the energy transfer efficiency. For example, if you use a biosensor, then you have to fit the Becker formula, which we call it as, if you fit for 2-component analysis, the first fast component is quenched lifetime, and the slow component, which is tau2, is considered as donor alone. So you can also use that kind of approach to calculate the energy transfer efficiency using the lifetime technique. All right, so as I mentioned before, tryptophan is it's a neurodegeneration. It is mainly the cancer center people are very much interested to investigate the tryptophan during cancer progression. There is a kynurenine pathway involved. So that is, there are a couple of professors are working in the medical school on the tryptophan kynurenine pathway in cancer progression. So tryptophan is an essential amino acid, is a precursor of niacin, which in turn is a precursor for NADPH. And tryptophan, known to be FRET pair, were investigated on the basis of NADPH interacting enzymes carrying tryptophan residues involved in the FRET during cellular metabolism. So we use the 740 nanometer as excitation. As I mentioned before, we are following the change in lifetime of the tryptophan during drug treatment or cancer. During the drug treatment, I show you how it changes the tryptophan value, how the energy transfer efficiency changes here. So as I mentioned, we hypothesized the NADPH interacting enzymes carrying the tryptophan residues are responsible for quenching the tryptophan. Here is the enzyme carried by the NADPH, number of enzymes, number of residues that is responsible for reducing the lifetime of the tryptophan. So here it is, it's a link capsule line. You can see the blue is the control, and with the different timing of toxorubicin treatment, you can see increase in energy transfer efficiency. The increased interaction of tryptophan or residues in protein enzymes with the NADPH with the increased mitochondrial OXPHOS activity. So this is the African American cancer cell line. At the bottom graph, you can see clearly we see the energy transfer efficiency. So how do you how do you say it is true? How do you demonstrate it is true what we are seeing? So we did some, as you know, the toxorubicin introduces the apoptosis or caspase-3 activity. So it takes time, say 1 hour. It is really apoptotic takes place 12 hours or 24 hours. So if we treat the drug and keep the cells for 24 hours, then we should see the energy transfer, the morphology of the cells changes, and there is no increase in energy transfer. And you see the blue color is the control, and we could not see any energy transfer efficiency. It clearly tells you that what we are observing, what we are seeing, the interaction, the residues which carries the NADPH reduces the tryptophan lifetime. It quenches the lifetime. All right. So I think I have 10, 15, almost 2 minutes or 3 minutes. Now I am going to introduce the machine learning, what we do or what we did. As you know, the lifetime is one of the wonderful technique. I have been in the lifetime working and building the systems for a long time. There are a number of parameters comes in. Since I started the lifetime work, we are using only one data. For example, quantity donor, and then you calculate the energy transfer efficiency or redox ratio for FLIRR, 2 value, bound enzyme and bound FAD, just 2 value. You calculate the redox ratio. But there are a number of parameters involved the biological process. The lifetime spits out number of data. How do you use all those information to find out, to improve the sensitivity of the measurements or the drug response? That's what we did. The student did fantastic work using the machine learning approach. So there are a number of parameters involved, A1, tau1, tau2, photon counts, etc. And you see we created the heat map. We use the 14 lifetime parameters, which includes all the tau1, tau2, and photons. All those parameters we include. This is called heat map, which is a Pearson correlation matrix, which assesses the reasonable applying PCA, which is principal component analysis. We use the autoencoder approach, which is not supervised. It's unsupervised approach. That is the right way to do for our work. And the heat map, you can correlate the very bright, tells you better correlation. Other area is not that much correlated. You can correlate all those data. This on the right side is the correlation coefficient. You can clearly see there are a number of ways you can represent the data. So the autoencoder features non-linear combination of input variables. The principal components, the PCA, is linear combinations of input variables. So how it is advantages for our work. So this is the FLIRR on the left side. You see the FLIRR so much crowded. So this is the PCA. You see that the broken line shows the PCA results, which is very much cleared. And the ML features, machine learning features, clearly shows the drug response within 30 seconds. We don't have, or 20 seconds. You don't have to wait for 20 minutes. We don't have to wait for 60 minutes or 1 hour. So the sensitivity of reading the material or reading the information, drug response, is quicker or clear. And the KS value statistics, which is involved in the machine learning process, you can clearly see the FLIRR is at the bottom, and at the red is the ML feature, machine learning features. It clearly shows more sensitivity than the FLIRR, which we use to 2 data, than using the 14 data points, which is provided by the FLIM data analysis. Of course, everything was extracted from SPC image of biochemical data. We just extract the data and then use the machine learning. It is not one data, it is thousands or hundreds of data was used. Of course, it's hundreds of data is required for machine learning. Otherwise, it will not give you any data. And here is the HeLa cells. The same way you can see the sensitivity of this. Here, the FLIRR is green color here at the bottom. The red broken lines is the machine learning approach.
Terry,
I am done.
Okay,
I am done.
Okay, do you want to Okay, perfect. I do have a question for you in the chat. It's from Trung saying, thank you for a great talk. I love the idea of using single wavelength for NADH and FAD excitation, a 780 nanometer fiber laser instead of 800 for this application.
We selected 800 nanometer because of all our correlation works good. 800 nanometer is very good signal we see for FAD. You can use 780 nanometer fiber laser for this work. It's only 20 nanometers difference. The FAD signal, if you get enough photons, you can get it. But again, the single wavelength selection also varies depending on whether you use the tissue or single cell or cells, monolayer cells. So you have to try to find out appropriate single wavelength so that you get enough NADH photons and enough FAD signal so that you can fit appropriately.
And another question that I have for you, you were talking about long timescales for imaging. Do you see any damage occurring to the mitochondria with the single wavelength?
We did not do long time recording. Maybe less time recording, I would say. It's mainly 30 seconds. We have not tested whether the mitochondria is affected or not, but we don't see any, the cell. We can do time-lapse imaging for a long time. We did not see any damage that much, but we have not tested. We collect only 30 seconds for NADH and FAD data collection.
And then one last question. Holly wants to know your secret for not aging. She said you, Peter, and Karsten all look the same. Maybe all the people working on lifetime imaging, they have a secret.
Yeah, maybe. Yeah, that is true.
Okay, so thank you, Perry, for a fantastic talk. I again invite everybody to join him in the Q&A after Stephen's talk. Thank you, Perry.
Thank you.